Glycolysis takes place in the cytoplasm. It is an anaerobic process, which means it can take place without oxygen. It is a breakup of glucose 6-carbon ring into two 3-carbon molecules called pyruvic acids or pyruvate molecules. Glucose is broken down into 2 PGALs, also known as phosphoglyceraldehyde. This requires 2 ATPs. Then 2 PGALs are converted to 2 pyruvates. This creates 4 ATPs and 2 NADHs. At the end, glycolysis produces 4 ATPs and 2 NADH, but uses 2 ATPs in the process, for a net of 2 ATP and 2 NADH. The Krebs cycle undergoes the aerobic process, which means it needs oxygen to function. It occurs in the inner membrane of the mitochondria. It takes carbon-rich pyruvate molecules and reworks them to create another 2 ATP per glucose plus some energy. One of the pyruvates is oxidized, which means it is combined with oxygen. Then one of the carbons bonds with oxygen and leaves the cell as CO2. The 2 carbon compounds that are left are called acetyl-coenzyme A or acetyl-CoA. Another NAD+ comes along and picks up a hydrogen and becomes NADH. 2 pyruvates create another 2 molecules of NADH, making it a total of 4 NADH. Enzymes bring together a phosphate with an ADP to create another ATP for each pyruvate. Enzymes also help join the acetyl-CoA in a 4-carbon molecule called oxaloacetic acid together, and they form a 6-carbon molecule called citric acid. Citric acid is then oxidized over a number of steps, cutting carbons off to eventually get back to oxaloacetic acid to start the process all over again. As the carbons get cut off, there are leftovers in the form of CO2. Each time a carbon comes off of the citric acid, some energy is made, but it's not ATP. It's stored in a whole different molecular package. NADs and FADs pick up hydrogen and energized electrons from each pyruvate, which in effect charges them up. The addition of hydrogen turns them into NADH and FADH2. Each pyruvate has 3 NADHs and 1 FADH2 per cycle. Since each glucose has broken down into 2 pyruvates, each glucose molecule can produce 6 NADHs and 2 FADH2s. The main purpose of the Krebs cycle is to make these powerhouses for the next and final step, the electron transport chain. The electron transport chain is an aerobic process, which means it needs oxygen to function. In the electron transport chain, NADH and FADH2 electrons provide the energy that will work as a pump along a chain of channel proteins across the inner membrane of the mitochondria where the Krebs cycle occurred. These proteins will swap these electrons to send hydrogen protons from inside the very center of the mitochondria across its inner membrane to the outer compartment of the mitochondria. Once they're out, the protons want to get back to the other side of the inner membrane because there's a lot of other protons out there and they seek a balance. Protons are allowed back in through a special protein called ATP synthase, and the energy of the proton flow drives this crazy spinning mechanism that squeezes some ADP and some phosphates together to form ATP.